Hello, in this video we're going to be talking about the control factors of the cell cycle. So let's not waste any of your time. Let's get right to it. Now when we're talking control factors of the cell cycle, the things that we want to emphasize are the three major checkpoints. We want to emphasize what is happening there, what they're looking for, what they're checking for. But we also want to talk about the positive and negative regulators. We want to talk about the inhibitors. We just want to talk about everything that has a purpose of making sure that everything is going well at each stage. That way, the decision can be made if we're going to advance the cell cycle or not. But it's a process. Proteins are working to get everything done. The three checkpoints. There's one in G1 that I mentioned back in the G1 video. There is a checkpoint in G2. Okay, and then there's one in the middle of mitosis, right before the anaphase stage. But we'll talk about each of those individually. Now, the idea of how long it takes for a cell to divide, man, there's a lot of factors that go into that. If a cell, especially if it's an embryo cell, and just doesn't have a lot of stuff going on yet, I mean, it can breeze right through in a couple of hours. But if it's a cell that's a normal adult healthy cell, it can work its way through. Maybe it comes across a situation to where it needs to take a peek at something that looks bad and then work on getting that fixed, a couple hours maybe. Or some cells, because they're so specialized that they may be waiting for a certain condition. They could be in that G0 state for a long time. Excuse me. Now I mentioned the three checkpoints. The first checkpoint, the one in G1, it's just assessing how the cell growth is going. Okay, how's the cell development going? Is everything going well? Is everything in place? Everything is just working its way. G2, we want to make sure that the chromosomes are all set. The spindle fibers are looking good. I shouldn't have said spindle fibers. That's checkpoint 3, the M one. But just everything is going well as we advance towards mitosis. And of course, the third checkpoint, the one in the middle of the M phase mitosis, I mean, that's the one that's checking to make sure that the spindle fibers are in place. And it's mostly looking at the importance of making sure it's all good, because once those chromosomes separate, man, that's irreversible. So you just got to make sure that everything is going well. Now, when we're talking environmental factors, external factors, I should say, we're not talking external factors as far as outside of the body. Okay, we're talking external factors in relationship to the cell, which means around it. Okay, so we're talking the cell, and then everything outside of that would be the external factors. And I mentioned this briefly in an earlier video, but say a cell is in the process of cell cycle, and the cell next to it sends out the alert that it's been damaged by bacteria. Okay, that's going to affect if the cell goes through the cell cycle or not. The cell may decide, whoa, we got to wait for that situation to get cleaned up before we look at going any further. It could just be the brain giving a signal out. No, not right now. We got something going on. Maybe the T cells are around. Immune system is busy doing something. There's a lot of things that could come into play that the cell cycle would be halted. Wouldn't be thrown away, but it's just halted until things get to be a better situation. And of course, G1 is the big one about that. If there's environmental factors are going to affect one of the checkpoints, and G1 will be that checkpoint. So at the G1 checkpoint, the G1 checkpoint is just simply going to decide whether we're going to go on and divide or not. Again, we're checking to make sure everything around us is good, making sure everything internally is good, that all conditions are in play. Things like the growth factors, are we growing well? Are we developing well? Is everything going on in that matter? If it meets it, great. If not, we just need to pause until that gets cleaned up. Or if we need to go into the G0 stage and await further instructions about developing. But G1 is just all about checking to make sure that everything is going well, both inside, internally, outside, externally. Really focused on that deep analysis of our current situation. So the G2 checkpoint, okay, now the S phase has occurred and the DNA has been replicated. So now we're checking to make sure that that's okay. Is there any problems, any concerns with the DNA? The proteins report anything? Do we need to stop so the replication issues can be dealt with? Avoid mutations, avoid changes that could throw off everything. If all is going well, great, we'll proceed. If not, then we are going to just do whatever is necessary to get it fixed. Okay, at the metaphase checkpoint, also known as the spindle fiber checkpoint, that's really what it's looking at. It really has to make sure that everything with that separation of the sister chromatids is in place. Because as I mentioned earlier, once that anaphase snap has occurred, I know the word snap with Marvel is pretty possible. That's a different thing. We're talking separation. Once that happens, that's, as I mentioned, that's irreversible. You can't put that back together. So you really got to make sure that has occurred. So if you're looking at a multiple choice question about the M checkpoint, you're really looking for an answer that talks about spindle fiber analysis, making sure that the separation to the centroids is good, to the separation to the attachment of the kinetogears gears, and the centrosome is in good shape. If not, because one thing that this is trying to avoid is a disproportionate number of chromosomes in each of the cells. That's a bad thing. They don't separate with the right amount in each cell. It creates non-disjunction, and you may think, what's a big deal? That's what Down syndrome is about. It's about not inappropriate, but incorrect separating of the chromosomes. This one's got so many, this one's got so many, and it's not the half number that it should be. All right, the GO phase I mentioned a couple of times. This is that temporary little place to where the cell can just call timeout. It's not a timed timeout. Length does not matter. Duration does not matter. What matters is getting everything right. So the cell can just go into here. If it gets into the G0, I keep saying G0, and eventually looks like it's not going to get fixed, whatever the issue is, then we can go into cell death, apostasis, and take care of it that way. But again, the duration that a cell spends in G0 is indeterminate, and it's very different, a lot of varieties, and just making sure everything gets done. All right, so now we're going to begin to look at the regulators, because not only do we have these checkpoints, but these checkpoints are ran by molecules. We've got your positive regulation molecules, and those are designed to promote the moving on to the next step. The other ones, the negative regulators, those are molecules that say, no, this ain't happening. This is not right. This is bad. It's a bad system, bad setup. We got to shut this down at this point in time. Okay. Also, so kind of looking at positive regulators. You can see there by the diagram that different types of those cyclin, the CDKs, those enzymes, those proteins, different types show up at different of the different checkpoints and different processes. You got your D's and E's and A's and B's, but it's just that idea that binding to the molecule, really checking it out, looking it over. And again, you can see the written text there, but they're just a major part of what's happening at these various checkpoints. Okay, so again, positive regulators. Now, these are obviously proteins that I mentioned, and they phosphorylize other molecules, so it means that they change them. They change them to CDKs. The CDKs are the main molecule that does this. So if you're looking for a question about which of these choices is a positive regulator, the CDK is what you're looking for. But they're super important in deciding if the cell's going to move on to the next stage or not, or do we need to stop and wait and take care of that. Okay. Now, with positive regulators, there are also molecules. Speaking of just checkpoints, checking. And not only the positive regulators do their part, but there are molecules or things that are designed to monitor and to inhibit, if necessary, the CDKs or the positive regulators. They can be, shut it down, repair, fix it, whatever. Yeah, just the idea of looking over what I had here to make sure I wasn't missing anything. But the idea of these inhibitors, they just simply bind to it and change it. If we're talking enzymes here, we know that from past talk that enzymes, if you bind to the active site, then you can fit right in there. But if you bind to the allosteric site or somewhere other than the active site, then you can alter the shape of that enzyme and where it can't do what it needs to do. Now the negative regulators, the regulators that are designed to stop the process, the best known one of those is P53, and we've looked at P21. These are designed to kind of just shut down the process. They can, whatever to detect there's some damage in the DNA, something is wrong somewhere. But these negative regulators, once they're out there, they're shutting down the process. That P53, as you can see there, I mean, it's an important one. That G1 checkpoint, but it's the idea. It's the protein that can determine if the DNA is damaged or not. Okay, it can recruit proteins to come in and fix on that and work on that. It's the one that decides, hey, if we're going to go into GO or not, or do we just need to shut it down because it's just too bad off and kill the cell. Now, between these two types of regulators, a lot of this work was determined or found during research on cancer cells, because they divide so fast and so rapidly, and you can kind of see what's there. But again, P53, P21, the knowledge of that is going to come out of cancer research. Well, that's our video over control factors of the cell cycle. When we come back next time, we're going to be talking about cancer and the cell cycle. We'll see you then.